Hi, everyone, and welcome to episode 300 of your Tick Bootcamp podcast. The title of today's interview is Ahead of the Curve, an interview with Lyme pioneer, Dr. Alan McDonald. My name is Richard Johansson.
And I'm Matt Sabatello.
Folks, we want to thank you for following us for 300 episodes. This is a benchmark episode for us. And for this special episode, we invited one of our favorite people in the entire Lyme disease community, pioneer Dr. Alan McDonald.
And you're going to be blown away by the recent research done over the last year by Dr. McDonald. He's had some recent discoveries about Lewy body dementia and how the scaffolding of these Lewy bodies is actually the Lyme DNA itself. He's also done some research regarding sequestering, which is how the Lyme bacteria itself actually uses your immune response to protect the spirochete. We talk about liver infections and how 20% almost of the acute early Lyme patients have liver issues pertaining to Lyme disease that affect detox. We talk about leukemia and how leukemia is being found now to be caused by Lyme disease. We talk about glioblastomas and brain cancer that we talked about on our previous podcast with Dr. McDonald, and some groundbreaking new discoveries there as well. And finally, we talk about the connection between Lyme disease and suicide and how important it is to better understand this connection.
So, folks, we're really excited to reintroduce to you the Lyme pioneer who is always ahead of the curve, Dr. Alan McDonald. Dr. Alan McDonald, one of the Tick Bootcamp favorites, quite frankly. This is episode 300 of our podcast, so it's one of our benchmark podcasts, and we wanted to have a special guest. And Dr. McDonald, you're the special guy. So how does it feel to be the special 300th episode guest of the Tick Bootcamp podcast?
Well, it's a privilege. It really is the second time I've gotten to be on the program. That's much appreciated.
Well, we're really excited to have you, and thank you for reminding our folks that you were actually profiled in episode 171 of the Tick Bootcamp podcast. So if folks want to know more about you, because we're not going to spend a lot of time today giving folks your background, because that is available in episode 171. So again, thank you, Dr. McDonald, for taking now a second window of time out of your life and out of your family's life to meet with us and to speak with us. So Dr. McDonald, we're going to talk with you about, if you would be kind enough, talking to you about 6 different topics today. And I just want to highlight those for you. First thing we want to talk to you about is the research you're doing on Lewy body dementia and some of the advances that you've made there. We want to speak about Lyme negative serology and some of the work that you're doing specifically on sequestration. I'm going to talk to you about the liver and some of the work that you've done with the liver, and specifically liver damage that's done during the acute phase of Lyme disease and the impact that that's having on folks in the community. The fourth issue we want to talk with you about is some of the research you're doing with CLL and leukemia and some of the challenges that Lyme disease is creating in that window. We're going to sort of do a follow-up with you on the research you're doing with glioblastomas, because you did give us some really powerful insights last time, but we know you've made some advances on your research and some new findings. And then the last thing, which of course is really important for folks in the Lyme community, not that any of these are less important, but you've done some research with a person who had unfortunately committed suicide, and you made some findings there. So Dr. McDonald, let's begin at the top if we could, and let's talk about Lewy body dementia and the impact that your research is having in that arena. And of course, folks know that you've been doing this research for close to 40 years. You're one of the Lyme pioneers, and a lot of the research that you were doing back in the day was actually ignored. And now you've gone from being the outsider who couldn't get his research published to now being published in mainstream research. In mainstream research journals. So let's talk about first Lewy body dementia, if we could.
I published just recently a major discovery in 4 patients who had Lewy body dementia and whose autopsy brains were donated to the Paul Duray Research Foundation for our studies. And we found that the Lewy body, which is a very small red staining structure that's sometimes hard to see inside of neurons. It's very small, but it is associated with neuron death and with progressive dementia. I found that structure contains DNA. Now, up till this point, it was always thought that the Lewy body was a protein-related structure, and that the protein inside the Lewy body was alpha-synuclein. And they went even further and said that because the Lewy body stains with alpha-synuclein, the alpha-synuclein protein there is toxic and it's causing the damage. My research has overturned that, turned it on its head, and I've discovered that alpha-synuclein protein, which is healthy in normal brain nerve cells and is present in increased amounts in Lewy body brains inside the Lewy body, is actually a DNA binding protein. And so its mission statement is to bind to DNA. And that means that because it stains very darkly with this protein, the target is DNA inside the Lewy body. And I went even further and I used my DNA probes for Borrelia, and I found that the DNA is the Borrelia DNA in 4 patients. So that's a major discovery, because Lewy body dementia is the second most common dementia after Alzheimer's disease. And indeed, 1/3 of Alzheimer's patients have a Lewy body component. And my grandfather's Lewy body dementia got me started in research. I wanted to find out what I could about that. So, his disease was marked by loss of his intellectual function and very disturbing hallucinations, like flying monkeys and giant insects in the apartment that he lived in. And those are scary type hallucinations, which mark the end of Lewy body dementia. Of course, Robin Williams had Lewy body dementia, and we all felt terrible when he passed away. So there are, Ted Turner has Lewy body dementia. So a lot of people in the news have Lewy body dementia. The wonderful thing about it, if there is a wonderful thing, is it's not related to amyloid at all. Alzheimer's is a so-called amyloid toxic disease. Lewy body is a so-called alpha-synuclein toxic disease. So what I found is an independent pathway from infection of the brain with Borrelia, namely Lewy body dementia, that gets us to prove that that infection causes the dementia of Lewy bodies. Right now, there's a great controversy about amyloid and toxic, and whether the original paper was fraudulent, and that most of the amyloid research in the last 30 years has been misplaced. So there'll still be a discussion about that, but Alzheimer's disease is not necessarily connected in a substantial way to a Lewy body problem. Alzheimer's disease is a separate problem. In the final event, the nerves die.
So Dr. McDonald, let me ask you to pause there for a second. I want to walk it back a little bit so that we can give folks a context here for our community. So our community, of course, as you know, are folks who are, in most cases, managing chronic Lyme disease and the family members and supporters of the people who are on that journey. Right. And I'd like to sort of like give this a sort of a higher level first, and then we'll walk it down into some of the great research you're doing and some of the findings that you're making. So we have a lot of folks who are suffering with neurological Lyme symptoms. Sometimes it's brain fog. Sometimes it's even worse. Matt, for example, had very, very severe neurological challenges. And I guess the first high level question that I have for you is are your findings and all the research that you're doing giving you insight into what long-term impacts folks could suffer from if they are suffering from neurological Lyme disease, and why is that significant in the context of people like Robin Williams and Ted Turner and other people who are dealing with or have dealt with these challenges?
Well, the last thing I want to do is to scare all the Lyme community into thinking that they're going to have an end of life Alzheimer or end of life Lewy body consequence. I want to find an optimistic pathway so that understanding the brain infection, how it works, and why it causes the brain to be scrambled can be diagnosed early and treated with antibiotics, and then those problems that are the result of untreated, chronic, over decades infection of the brain can be stopped in its tracks. Now, in the worst case, after Lyme disease has done its maximum disease damage, in the brain, it has destroyed neurons. The neurons are gone. That's the worst case. In the early case, the neurons are just irritated and inflamed and still alive, and the spirochetes haven't killed the neurons. So we want to intervene at the stage where the spirochetes are in contact with brain neurons and other brain cells and get them to be eradicated with proper therapy. And so that's the incentive of this, is to get the disease diagnosed properly and get it diagnosed early and get it treated early so that it can be eradicated.
So the good news is, Dr. McDonald, if we are diagnosed early and we are treated early, then these challenges are not challenges that we're going to need to face. But that's why early diagnosis, competent diagnosis, and early intervention is really important. Is that your premise?
Yeah, but the problem is that the political community demands antibody positive results to confirm a suspicion of Lyme disease in any part of the body. And if you don't have a positive antibody test that satisfies the skeptics, then they'll tell you that you don't have it, you have something else, you have maybe a post-Lyme situation, or you have something that's a cross reaction of false positive. So those are stumbling blocks. And in part 2 of my research, which we're going to talk about, I'm going to explain why you can have robust, active, living spirochetes in your system, in your tissue, in your blood, but these will be invisible because the spirochetes absorb all of the antibody to their surface, and it's nothing left for the machine to test in the serum. That's diabolical. But that, when you understand that that's going on, we replace all of the current ELISA tests with a better test to find antibody-coated spirochetes or biofilms, and then use that as your diagnosis. Therefore, you don't have to be held back by a bad technology, which is doomed biologically because the spirochete has outwitted us, absorbed all the antibody onto its surface, and leaves nothing left for the machine to find. So your blood tests, of course, are going to be negative.
Dr. McDonald, we're going to get there because the next part of our conversation is going to be all about Lyme negative serology and how smart the spirochetes are in regard to their ability to hide from antibodies and the sequestering term, right? So we're really excited to get to that and share with our listeners what you've learned and how we can overcome that part of our journey. But I want to go back to the Lewy body dementia case here, right? Because, you know, sometimes we're talking about this and I'm questioning a lot of things because I'm not as brilliant as you, right? So I want to just break it down to make sure I understand exactly what you're saying. So Lewy body dementia, how I understand it, Dr. McDonald, is that a protein or a deposit or a mass forms in the brain. And that mass
inside the nerve cells.
Inside the nerve cells.
Inside the actual nerve cells. It has to be inside the nerve cells to count.
Okay.
It starts out by getting inside the nerve cell, and it makes a glob that's about the size of the nucleus of the nerve cell. And it doesn't stain blue, it stains red. And red staining stuff is thought to be protein. And so the protein that they worry about is the alpha-synuclein toxic protein, so-called toxic. Okay. And that doesn't belong in a healthy nerve.
Gotcha. So when you're doing your testing and you're staining your samples and you see red, not blue, that's indicative of a protein when you're doing an analysis of a brain tissue or a brain sample. And when it's always historically been believed that these globs or these proteins or these masses inside the nerve cells had this substance called alpha-synuclein, which is, I think, also called ASN, right?
Synuclein, yeah.
The nuclein, alpha-synuclein. And this alpha-synuclein, this ASN, was always believed to be the driving force behind this dementia, because when you have this ASN inside of a nerve cell in this glob, then it causes this dementia where you have deterioration of your brain, thinking, movement, behavior, mood, et cetera. Correct? Is that so far accurate?
Yeah, it's something that happens in Parkinson's disease all the time. Now, remember that healthy nerves have healthy alpha-synuclein, and Parkinson's Lewy bodies have so-called toxic, chemically modified alpha-synuclein. And so those are two different things to keep straight.
Right.
All alpha-synucleins are not bad. If you get the healthy one, which you're born with and healthy nerves have it, that's fine. If you get the so-called toxic one, then you're in trouble.
But are there really toxic ones? Because what I heard you say earlier, Dr. McDonald, is that we may be mistakenly identifying the ASN inside of these globs as being the root cause of dementia. But it sounds like they're just binding to this DNA, which is their mission statement, which is the DNA of Lyme disease or Borrelia-Burgdorferi, which may be the deeper root cause. Is that what I heard you say earlier? I just want to make sure I understood that part correctly.
You're absolutely brilliant. Right. Absolutely brilliant. The glob is actually a glob of DNA. And so because it is a glob, it doesn't have a membrane around it. It's just a glob. It doesn't belong in the cytoplasm. And it has a lot of alpha-synuclein binding to it. And that makes it a Lewy body. But the alpha-synuclein that is in the textbooks, it's supposed to be a toxic one. What I'm saying is it may not be toxic at all. It may be doing its duty because it's binding to the infected DNA in the glob. And the infection DNA is killing the nerve, not the alpha-synuclein.
So mainstream medicine right now believes there's healthy ASN and toxic ASN.
Yes.
But your studies, your research, your analysis of these samples of these brains are now showing that ASN is just binding to this Lyme disease DNA, and it's doing what it was designed to do. It's a God-given purpose, as you call it, right? So you're arguing that based on your studies, ASN may not be toxic at all in any case. It may just be doing what it was designed to do. And Lyme disease is at the root cause of Lewy body dementia, correct?
That's exactly right. Now let's step aside a little bit and take one look at amyloid. Amyloid in Alzheimer's disease, according to the dominant theory, is toxic amyloid, right? The amyloid is toxic. Well, Harvard has shown that amyloid in Alzheimer plaques is actually an antibiotic, good, beneficial amyloid. It's not toxic at all. And this furor over the fraud in Alzheimer amyloid research really shakes the amyloid people to their roots, because a lot of what they have been looking at with bias may be false. Just like alpha-synuclein may not be toxic, amyloid may not be toxic. It's a major coup. Change in thinking.
Well, I think this is important because sometimes we get set in our ways and people are getting sick and they're declining and they're dying from things. And if we start to understand better what's going on, we can maybe treat these patients, right? Because if we catch them early enough, to Rich's point, we might be able to treat them and get to the root, which is the Lyme disease, and maybe get these patients better, right? So, we know your testing, your fish testing and all of your stains are really cutting edge and brilliant. And we know you've consulted with Yale and a ton of other universities still to this day in retirement. But in one of your studies, we have your study in front of us, which was brilliant. I mean, it was just published, right, on August 10th of this year, 2022, all that microbial DNA, globular, liquid, crystal-like deposits inside Lewy bodies in 4 Lewy body dementia patients, as you talked about earlier. And it's really deep. And Rich and I spent days going through it and trying to take notes, and we still don't fully understand it. So we're trying to break it down for our listeners here to understand how deep Lyme disease can be beyond just, as you noted, some joint pain, right? So when you did these studies, you found, so you had these brains autopsied and they demonstrated Borrelia spirochetes, Borrelia specific proteins, and Borrelia specific DNA inside of the Lewy bodies, which attracted the ASN. And again, that was, we believe, the root cause, the spirochetes. But what do you mean by Borrelia specific proteins, Borrelia specific DNA, and Borrelia spirochetes? Is it possible that there's an active, fully functioning spirochete, but then almost like a remnant? Because we've heard studies from like LymeDisease.org and coming out of other universities that remnants of spirochetes can cause people to be sick as well. And we don't really fully understand what that means. So can you explain to our listeners what the difference between a spirochete, a Borrelia-specific protein, and a Borrelia-specific DNA identification inside these Lewy bodies actually means to our listeners?
Yes, the spirochetes that are in the body may be alive for a period of time and then may die. And at that point, then they can disintegrate, and all of the chemicals that are in the living spirochete can be gathered into a glob so that the dead spirochete DNA forms the glob in the Lewy body. It can also be in a biofilm, because biofilm is a mixture of live and dead, and most of the protective goo in a biofilm is derived from once living but now dead spirochetes. And biofilms are a marker of chronic infection, but they have a mixture of live Borrelia and dead Borrelia. And the live component we have ways of looking at it with DNA and the dead component. We can use the same DNA probes to prove that the DNA is also still there. The proteins are a wrapper for the DNA when in a living spirochete. So you have the proteins on the outside, the proteins on the inside, and then your living DNA. But when they die off, you'll have a remnant, and the remnant can form the glob in the Lewy body case.
So these remnants in the Lewy body case can be part of the glob, but also active spirochetes and also biofilms, right? So they don't discriminate any of these forms. But I guess the bigger question, in specific to Lewy body and specific to these proteins, but also broader picture, can these remnants, these dead spirochetes, these non-active, non-alive Lyme bacteria that for whatever reason your body is not able to process out or detox out, can they be harmful and can they cause damage? And it sounds like in this case, yes, because they're causing the Lewy body dementia by being the root cause of these globs or masses of protein inside the nerve cells in the brain. Is that correct?
Yes, well, there are two areas where the glob can wind up. First, it starts in the cytoplasm. And at that time, the healthy nerve nucleus is separate. So the compartment is cytoplasmic for Lewy body, and then the nucleus with the normal human DNA. Over time, the DNA that's in the cytoplasm can work its way into the nucleus. And once it gets into the nucleus, it can contaminate the healthy human DNA and lead to nerve death. So as long as the DNA of the Borrelia in the nerve, in the cytoplasm, stays in the cytoplasm, the nerve cell has a chance to stay alive. When the DNA gets into the nucleus and then causes the nucleus to die, then the whole nerve dies and you have an empty space in your brain, which used to be a space where a nerve cell lived. And as the disease progresses, you get more and more of these empty spaces. You can see them when you do the microscopic study, and you can see, well, there used to be a nerve inside that round space, and now it's empty. Or you can see there's a round space with kind of a decrepit nerve that isn't healthy looking and is starting to break up, and you can track it under the microscope. So nerve cell death can be tracked under the microscope.
And this is really brilliant information. So you're saying a dead Lyme bacteria, right? So, these remnants or these globs of Lyme disease, if they're inside the nucleus, they can lead to nerve death, even though they're already dead, right? So is that accurate? Okay.
Yes, because the human DNA doesn't coexist peacefully with other infection DNA. We're designed to keep it out, and we don't want to have any extra DNA from some infection getting into our DNA compartment. That's hallowed ground.
Right.
You don't want to have infection DNA in your nucleus. Viruses that kill nerves get into the nucleus and kill by destroying the structure of the DNA in the nucleus. Rabies does that all the time. So, cell death is a consequence of DNA turning up in cells and sites like the nucleus of the cell or cytoplasm of the cell where it does not belong.
So let's talk about what we understand in the Lyme community to be detox, right? When our bodies, either through herbal therapy, antibiotics, our own immune system, et cetera, starts to kill off the spirochetes, whether it's in blood or in our brain tissue, et cetera. When the bacteria dies, we've heard of the Herxheimer reaction, and your body you have to detox and you have to open up your drainage pathways, right? But in this case, it sounds like when these remnants of the Lyme bacteria exist, they can still cause damage if they're inside the nucleus. So how does the body rid dead spirochetes or dead Lyme bacteria, these globs in the brain? Is that still an immune response? Is that a detox response? Is it different because it's inside the cell and it's harder to get the remnants outside of the cell? Like, walk us through that process there of what's supposed to happen in a healthy body when the Lyme bacteria dies inside the brain, inside a cell, and how we can get rid of it.
Okay, well, the sequence of events is that the spirochete comes from the tick, it gets into the body, it gets into the blood, it gets into the brain, and then it gets into the neuron cells, right? So it moves from all these different compartments, jumping from one to the other. Antibiotics and detox can help with the spirochetes that have not died yet. As far as what what you can do with a glob of DNA from a dead spirochete, I'm not really sure, but the goal is to get the therapy working before the thing dies inside the neuron. After it dies inside the neuron, then you may just have to kiss that up to God and survive very well with one less neuron. Everyday healthy people on planet Earth are estimated to lose thousands of neurons from their brain just because of normal aging. So loss of neurons is not against physiology and biology. We're born with more neurons in our brain than we have when we die. So, we have progressive loss of neurons, and some of that is just natural. They don't live forever. Spirochetes accelerate neuron cell death. And because of that accelerated death, then you get, when the neurons are dead, then you have to make the best of what you have after that. You want to get in a detox before the neuron is killed and before the glob is there.
And when we speak about these globs, we're still referring to the globs which represent this mass or the protein inside the nerve cells, which are causing Lewy body dementia. Those are one and the same for the globs we're talking about with the dead remnant bacteria. They're not separate, correct? This is all still the globs of proteins in the brain that lead to Lewy body dementia, correct?
Partial correct. The globs have protein and they also have DNA. And the protein is designed to bind to the DNA. So without the DNA in the glob, the protein wouldn't attach to the glob, right?
Mm.
See, now you got it.
Gotcha.
Right? So you have a double package, right? And in my pictures from the paper, you can see that there's a picture of proteins in Lewy bodies. There's a picture of DNA in Lewy bodies. So there's both things going on. They say that there's probably 800 different chemical species inside a Lewy body. It's not just a couple of things. There's a lot of other things, and some of it may be cells that, cell products that the host makes, and some may be products of what the infection makes.
So talk to us more about your research study that dropped in August, right? Because this was really powerful and there's a lot of great information. And I know one of your major conclusions is that Borrelia or Lyme is really the scaffolding. That Lewy bodies are built upon a scaffolding of Borrelia or Lyme DNA, right? And that was something that was sort of groundbreaking in the study. How was it received by the community? And are any of your peers and fellow researchers and scientists reaching out to extend this research or collaborate or take it to the next level?
I've sent it to the Lewy body research teams around the country. I just emailed the directors of all the programs, and I sent the full manuscript to them. I got a reply from one director of Lewy body research in a medical center. I'm not going to mention who it is. I don't want that person to get in trouble, but all the other people just rejected it or didn't reply to it. The thing is that this will fundamentally change not only Lewy body research, but all of Parkinson's research, because they both are Lewy body diseases. So this is going to chip away at the Michael J. Fox Parkinson's Research Network community. It's going to chip away at the Stuart Udall endowed research programs in Parkinson's disease at major medical centers around the country, like Harvard and Hopkins and all those places. And it's going to fundamentally impact what's going on with that research too. And hopefully people will find value in it and start to look at the possibility that they've been missing infection DNA inside of Parkinson's infected or Parkinson's disease cells. There is something that's very, very delicious. It's called post-encephalitic Parkinson's disease. And it came up where they had an excess of Parkinson's cases after the big flu epidemics earlier in the last century. And they said that in fact, after you have a very bad encephalitis with a flu or a bad flu, some of those people will get Parkinson's, and the residual increase in number is noteworthy. There's enough of them get Parkinson's after having this infection that they've coined the term post-encephalitic Parkinson's. Isn't that beautiful? So it opens up a 12-lane expressway to truth, because infection and Lewy bodies and Parkinson's is already benchmarked and approved in the textbook as a real entity. So what I'm doing is I'm adding a little thing called Lyme disease to the infection preceding a Lewy body disease, namely Lewy body dementia, but it will also probably play out in Parkinson's disease because Lyme can probably have a Parkinson's type post-infection consequence. So you have a lot of bonus points here. The thing is that if people have invested all the time and money in the wrong direction for the past 20 years, they're not going to be happy about my research because they have to pay their tuition payments and make sure that they can continue to get the grants for stuff that they've been studying, which may not be relevant to the real cause. So it's a major threat to the research community because you have to retool everything in Lewy body of any type to get to approve my results.
What I find fascinating, though, Dr. McDonald, is that it's medically accepted in the world that bacteria in general can drive Parkinson's and Lewy body dementia. But when you start to say specifically Lyme disease, which almost 15% of the world population has been infected with, can be a contributing factor, they want nothing to do with you, right? So again, Lyme disease seems to be this controversial disease all around. And once again, you're clearly and definitively proving the connection, and people are just saying, that's crazy. We're not going to believe that. Just like they did to you 30 years ago, up until, the last couple of years when they started to realize, this guy's right. There's some real stuff here. But I do want you to break down Parkinson's, because we understand Lewy body dementia. You explain what that is simply for us. Can you explain what Parkinson's disease is and how it's different than Lewy body dementia? And then how your research and your studies are connecting Lyme disease as being a trigger to Parkinson's disease?
All right, well, the Lewy body in the nerve is essential for Parkinson's. The actual anatomy of which nerves get the Lewy body deposits separates Lewy body dementia from Parkinson's. In Parkinson's, it's in the substantia nigra at the base of the brain and lower areas of the cortex called the striatonigral cortex. It's a big mouthful, but it's not up on the top of the brain where we have a cortex. And in Lewy body dementia, the Lewy bodies are in the cortex region and may not be present at all down in the Parkinson's region of the brain. So there's different anatomies that are diseased. The only other, important thing to keep in mind is that many Parkinson's people will not get dementia. They'll just have the movement disorder and the shakes and the tremors and that's a movement disorder. Now there's a subset of Parkinson's that gets dementia, but not everybody with Parkinson's and the Lewy body is essential to Parkinson's will get dementia. The cortical part, the Lewy body up on the top of the brain, the Robin Williams type people will have dementia and maybe have a little bit of a shake. He was originally diagnosed with kind of a sort of a Parkinson's thing because he had some shakes, but it wasn't as major Parkinson's-like, but he had some Parkinson-like symptoms. So it was spread across the upper brain and the lower brain. And that's the anatomy that defines the differences between the lesions. Once you have the Lewy body inside the nerve, the rest of the biology is as I've described. You got the glob in the cytoplasm, you got the covering of the alpha-synuclein protein on the cytoplasmic glob, and the DNA eventually works its way into the nucleus, and then that kills the nerve cell, no matter where it is.
So really, it's all driven by Lewy body, whether it's Parkinson's or Lewy body dementia. It just depends on the geographic region in the brain at which these globs form and impact the person, whether it's just dementia, or I shouldn't say just, whether it's dementia with Lewy body dementia or the movement disorder with Parkinson's, but it's the same underlying, we'll call it the same underlying trigger for both conditions, correct?
Yeah, it's the action is in the nerve cell. That's where the action is. And everybody agrees that the nerve injury is the root cause of each of these diseases. And it's just what gets the nerve injury going and what keeps it going is where the research is.
And I do want to correct something I think I may have said incorrectly earlier. When you had said that it's accepted that I think viruses trigger Lewy body. Did you say bacteria as well, or just viruses? And your research is now introducing the concept of bacterial-driven Lewy body.
I'm not sure that viruses trigger Lewy body. I'm not saying it's impossible, but I think that Lewy body changes in post-encephalitic Parkinson's, which means post-influenza Parkinson's, are related in that specific definition that it's a viral illness that overwhelms the brain, and it's like a major flu, like the 1918 flu it's all viral. And then after that, there was this, Lewy body type consequence. So yes, viruses can do it, but you have to be sure you know what your viruses are, and you have to be sure you know what your bacteria are. You don't want to make a broad, sweeping statement. You want to be very, very, very specific. We're in the era of precision medicine. So we speak with precision, and we show pictures with precision, and we don't, confuse one category with another.
So I think you're saying which specific viruses can lead to it versus which specific bacteria, like Lyme disease, can lead to it as well, is what you're saying, correct?
Yes.
So walk us through what's next in your work with Lewy body dementia and all of the brain research you're doing specific to Parkinson's and Lewy body dementia, and your research that came out in August, before we move on to the next topic, which is equally as interesting, which is that Lyme negative serology and sequestering.
Well, unfortunately, I don't have access to any Lewy body Parkinson's type tissue. I don't have any Parkinson's in my collection of cases for study. It doesn't mean I can't get some, but right now I've been studying just the Lewy body dementia thing. And in order to get 4 cases, it took me 6 years. They're not standing on every street corner. And you have to be very lucky sometimes to get a brain autopsy that has a patient who was suffering from Lewy body. All of my patients had the 2 key features clinically, and that is that they had a demential decline, and they had very, very scary hallucinations, very scary, flying monkeys and giant insects and other things, very scary things. That's one of the things about Lewy body dementia, which people often have, is the scary, scary hallucinations. So when I get the chance to study Parkinson's, if I can get my hands on that, I will look just as I did with these other Lewy body categories. Right now, I haven't gotten a chance to do that.
So now let's talk about another really important topic in the Lyme community, Dr. McDonald, which is testing. Testing is a huge issue. And even within the folks in our community, there's a constant debate about whether or not you actually have Lyme disease if you do not test positively. In fact, I recently responded to a young woman who commented on one of our social media posts who said, get over it. If you don't test positive, you don't have Lyme disease and you should be looking somewhere else. And we had an interesting exchange with this woman. So now your research is now demonstrating, specifically your research with sequestration, is demonstrating why these tests are not working and why we need to create other types of tests. So can you share with folks what sequestration is and why that now gives us an understanding of why the traditional 2-tier test is failing folks in the Lyme community?
Yes. The antibody that the machine can see, and all of our tests for all intents and purposes are machine readings of serum, need to have enough antibody in the serum for the machine to detect it and produce a color reaction, which gives you a number and then gives you a result. Now, in addition to people who have low antibody levels, real disease, but have low antibody levels because they're on steroids or they're sick or they have cancer or they got a very advanced infection which has suppressed the immune system, or they have an immune deficiency, born with it, or they're a young pediatric patient, they haven't got the mature system going with the immune system. All of those things can terminate in a blood test which is flat out negative. And I started the blood test myself, or I was early involved in the blood test when it was using whole spirochetes smack dab placed on glass slides. And what we would do is we'd make little rounded areas of deposition of whole spirochetes on glass slides, and then take the patient's serum and layer the serum over and see if the fluorescent marker for bound patient antibody to spirochetes was bright enough to pass through dilutions from 1 to 8 to 1 to 256 to 1, 5, 12, and higher dilutions. That work was all done with a microscope and a single human person, me, looking at the fluorescence of the spirochetes that had bound the antibody and seeing if it's bright enough to see after you dilute it 256 or 512 or 1024 times. The higher the dilution that you would see would be the result of a positive test. Anything that was less than certain cutoff, usually it was 1 to 64 early, and then we made it 1 to 128 dilution. If you got past that, then you had a positive test. All right, now, we know then that spirochetes, live spirochetes, grown in the laboratory and then smacked onto glass slides will bind antibody. That's the way the test is. Right now, ELISAs use pulverized, vaporized almost proteins, but they don't use whole spirochetes anymore. So you never see the whole spirochetes. The number of opportunities for the thing to bind are much better on a whole spirochete than a little puree of protein in a well. Now, it took me about 40 years to figure out that this whole spirochete test, the immunofluorescence test, taking out the antibody from the serum applied to it and producing a fluorescent signal could also be going on in the body of people who have living spirochetes still circulating in their blood and in their tissue. Those spirochetes will bind Lyme-specific antibodies. Yes, Lyme-specific antibodies will bind to whole live spirochetes in people who are chronically infected. So it's not new science, it's rediscovery of old science. And because spirochetes are solid and serum is liquid, when you get your blood ready for testing, you have to clot it. And then the tube is centrifuged and there's a little separator, and the clot and the fibrin and all of the solid spirochetes will go down to the bottom of the tube. And the only thing left at the top is the serum, right? Now, if the spirochetes have already bound all of the antibody in the whole blood, which they're supposed to do, that reduces the amount of antibody left in the serum compartment significantly. That's trouble number one. Trouble number two is that there are biofilms in chronic infection, and biofilms are hundreds of spirochetes living and dead, and you don't have to have a live one anymore. You can just take the protein on the biofilm surface, and that's like a sponge, and it pulls antibodies onto its surface. And then the biofilms are also removed in the clot, put to the bottom of the tube, and pull all of those antibodies out of the blood so the machine can't see it. And then there's the tissue spirochetes and the tissue biofilms in chronically infected people, which is even huger than the stuff in the blood because tissue outnumbers blood by a certain amount. So all of the sponge removal on solid spirochetes, solid biofilms, delivered into the clot, never to be seen by the machine, leaves you with a little area of serum that doesn't have the antibody that was in whole blood. And we don't do whole blood testing, we do serum testing. So you're really out of luck because all the antibodies in your body wound up in the clot. And I can show in whole blood that spirochetes alive in whole blood, which are invisible, have bound Lyme-specific antibody and they glow in the dark. And biofilms in whole blood bind Lyme-specific antibodies and they glow in the dark. And all the stuff that glows in the dark is inaccessible to the machine. No longer in the serum, it's in the solid phase.
So our tests
that's pretty scary for all the reference labs, isn't it? All the labs that do all the testing, right? And do all of their quality controls for machine reading Lyme testing should be having some kind of a gastric distress now because they have done with a technology, they've perverted a technology, which sounds really slick, but is biologically fundamentally flawed. Because the spirochetes pull the antibody out of the serum and there's nothing left for the machine to read. Or the antibody is so low that the machine reads a low reading when it's actually all in the clot, attached to the spirochetes, attached to the biofilms, pulled out, or attached to the tissue, never even in the blood.
But we knew this, and yet we still decided to modernize, quote unquote, and automate, right? And just put our blinders on to the fact that this is something that's going to take the quality of the test and destroy it because it's cheaper and it's probably something that's worthwhile financially to do, and taking the human element out of looking at the whole blood. Is that kind of where this is all coming down?
You
to,
are
Dr. McDonald?
absolutely spot on. And you know, the medical industrial complex is going to have me killed for this. I may be found in a dumpster somewhere because I've discovered this. And you know, IGeneX is going to have to go into you know, a laundry and ironing shirts because they're not going to be able to make their mega millions unless they do a biologically correct test. And the machine test right now is biologically incorrect.
So let's expand upon this a little bit further, because how I understand antibodies working with pathogens in general is your immune system will generate an antibody response to a foreign substance. The antibodies will hunt down and target the pathogen, so in this case, the spirochete, and then the antibody will then go in and attack and kill it. But it sounds like what you're saying, Dr. McDonald, is in Lyme disease with spirochetes, the antibody will hunt it down, but instead of killing it in many cases, just coats the bacteria and doesn't kill it?
That's right. It's cloaking it. It's coating it. It's not killing it, unfortunately. And because there's lots of reasons why the killing stage doesn't happen in chronic Lyme disease. But, you know, there's something called complement protein. Complement protein is one of the things that punches holes in cells and kills them. You know, if the immune system targets a bacteria or a bad cell and it wants to have it killed, the complement proteins come in and they actually punch holes in the cell and kill it. Spirochetes are known, Borrelia spirochetes are known to be coated with complement proteins. So then the immune system will be, you know, flying over like a drone, and they'll look down and say, well, there's complement, that's not trouble. I'm not going to do anything to that. I'm going to pass it over, you know. And so the immune system is deceived by complement coated spirochetes, just like it's deceived by antibody-coated spirochetes. It's deceived. It is blinded because they're cloaked. Yes, cloaking is common.
And so cloaking is also what we call the sequestering, right? So,
well, no, sequestering is slightly different, but it's a type of sequestering. But yes, it is in one sense. But sequesters are, you know, the immune complexes of Schutzer, and you know, he reported those things back, where the antigen that's dissolved in the serum binds to the antibody and makes an immune complex. And until that is broken open, you can't detect the antibody anymore because the antibody is already bound to a circulating protein in the blood. And the only way you can find it is to bust it open, and then you have free, unbound Lyme antibody, which you can actually measure. So it's a little bit of a difference, yeah.
So sequestering actually means that the antibody is doing its job and breaking down the spirochete and killing it, but then it's forming into this little ball where it's now not being recognized via blood test. Is that what that means?
Not exactly,
No?
but you're in the right direction. The antibodies may not be effective in killing for separate reasons than sequestering. You know, in order for the antibody to do its killing, it has to have some help from its, you know, friendly neighborhood complement. And if the complement is disabled, then, you know, it can't kill anything. It just coats it, and it decorates it like frosting on the cake. And it goes around, and instead of killed, it's a sponge. And the sponge is pulling the stuff out of serum so that the machine can't see what's in the body, which is on the solid surfaces of the infected particles and biofilms and in the tissue. The sponges have pulled all of that out, and there's nothing in the serum that, you know, unless it's a huge, huge amount, then you miss all the ones that are biologically positive because it's been sponged out by these solid things.
But at some point, your immune system...
That is diabolical, isn't it?
That is evil, yes.
That is diabolical, isn't it?
It is very diabolical. But I mean, at some point, your immune system has to think, I don't see any more bad stuff. I don't see any more Lyme bacteria because it's coated in the antibody. So does your immune system eventually stop mounting an immune response to Lyme disease because Lyme is flourishing and going crazy in your body, but just coated or cloaked in antibodies. So your immune system thinks all is good. You're getting sicker and sicker and sicker because spirochetes are flourishing and they're just coated with antibodies, it sounds like. Is that true, or does the immune system never stop pumping out the antibodies?
It's much more complicated than that. Actually, spirochetes are dirty, dirty things. And during their lifetime, they're continually shedding proteins and stuff from their surface. And that is soluble. That's not solid anymore. It's shed from the surface of the spirochete. So if you look at a picture of a spirochete in in our laboratory setting, you'll see that you have the cylinder, and then you have a lot of debris, dots of shed protein. And those proteins can do bad things with antigen antibody reaction. So it's not the simple equation.
So your body's always pumping out these antibodies for the most part because it's responding to the shedded spirochete, and then it's constantly being absorbed by the spirochete itself to coat it.
Antibodies respond to foreign proteins.
Gotcha. So
they don't. Sometimes they respond to foreign DNA too, but mostly it's foreign proteins, and the foreign proteins are continually shed from living spirochetes into the body system, and so that creates a fog, an immune fog reaction. And
a sense of confusion.
That can cause something where you have more antigen than you have antibody, and it's called a prozone reaction. And a prozone reaction is one of the basic, medical school first year things you have to learn about. It's like antigen excess can produce immune testing negative results unless you dilute enough to get rid of the excess antigen so you can find the antibody. So antigen is like immunology 101, antigen excess, prozone. And so prozone is also probably in play in some people who have Lyme false negative testing.
But the antigen is the bad stuff, right? So the antigen is the spirochete or the shedded part of the spirochete, correct?
That's the bad proteins, right? Bad proteins of the spirochete.
And if that outweighs the antibody response from the immune system, then you're not going to test positive because the antigens outnumber the antibodies, you're saying, correct?
Yeah, I mean, what you hope is that the antigen stuck to the plastic well in your machine, right? That's what it is. It's antigen purposefully attached to the edge of the well, will not be outnumbered by the antigens that are circulating in the blood already. If you're outnumbered, you're outgunned. And if you're outgunned, then you're giving bad data to patients.
So in response to when we started this conversation, when people are telling us on our social media, if you don't have a positive test for Lyme disease, you don't have Lyme, you're crazy, move on. That's just 1000% inaccurate based on sequestering, based on this cloaking, which we now know is slightly different, based on this whole theory you just described to us, where if the antigens outnumber the antibodies, you're not going to test positive for antibodies. And those are three scientifically proven reasons why you would not respond positive to a Lyme test, even if you have Lyme disease, correct?
Exactly. And, there's more than that. If you have your immune system is fundamentally weak, fundamentally impaired, then you can't make it. If you have HIV, then your immune system's not geared up to do anything much for other infections. If you're a newborn and your immune system hasn't matured enough, you can't make enough antibodies to meet the cutoff. There's lots of biological weaknesses that can interfere with getting a positive test. In order to get a positive test, you have to have a healthy, properly functioning immune system. And Lyme disease is notorious for having immune deficiencies just due to the overwhelming effects of the infection alone. You don't have to have HIV. You don't have to have steroids. You don't have to have cancer chemotherapy. But if you have all of those on top of an immune system weakness because of the infection alone, then you're doubly vulnerable.
Right.
And that's what a lot of people who have chronic situations have. They're doubly vulnerable.
So now we have the immune component where if your immune system isn't functioning well, that's another reason why you wouldn't be generating antibodies. That's a fourth reason. And many of them can be mixed and matched together to result in a false negative test, right? But yes. I know we need to move. We have so many topics to discuss. And before we move on to our next topic of the liver infections and all that wild information we've learned from you about even acute Lyme disease, I want to ask one final question on this topic, Dr. McDonald, which is, I'm still fascinated and curious why. I think a lot of Lyme patients will be. Why do some people's bodies, and this is probably a very complex answer, but why do some people's bodies generate antibodies? The antibodies find the Lyme bacteria and actually kill it successfully, whereas others, like chronic Lyme patients, it doesn't kill the spirochete, but it just coats it. And then as a result, they don't get better and they continue to get sicker and sicker and sicker and end up with chronic Lyme disease. What's going on in these bodies where the antibodies in one case can kill the spirochete and the antibodies in another body cannot kill the spirochete?
That's very complicated, and that's like a PhD thesis on the immune system and the sides of the immune system that have the cells, the lymphocytes that actually work to kill, and the antibody antigen complexes, which then work with the macrophages, which work with the rest of the immune system to make plasma cells, which make antibodies. So it's not that antibodies alone are your pathway to health. You have to have a healthy cellular immune side, and then you have to have a good macrophage antigens presenting system working for you. And people who have a very healthy immune system have the chance to do a much better clinically than people whose immune system is not up to par. I guess that's the simplest way of saying it, that not everybody is immunologically equal. And so, you know, one of my tragedies is a child that died at the age of 19 months, and he had a overwhelming Lyme infection, and his, you know, blood testing was negative. But there was a clue, and that was that the pediatrician had given him an immunization, you know, Prevnar, and the immunization didn't take because his immune system wasn't working right, right? So they gave him a second Prevnar immune, inoculation, and that didn't take. And then he comes up with negative Lyme testing. Well, the Lyme testing was negative because the immune system that didn't work for his Prevnar immunization wasn't working for his Lyme infection either. And, it's like, that's basic stuff. It's like, okay, well, if immune system A reaction is flawed because you can't respond to the vaccination, then everything else that requires the immune system to work may also be flawed.
Well, this is a really interesting topic because a large percentage of the chronic Lyme community is also diagnosed with one or several autoimmune diseases, and myself included, right? I think now the number is up to 3 suspected autoimmune diseases that I've had that I've been able to overcome most recently with some treatment. But do you think there's a correlation in people that develop chronic Lyme that have immune issues underlying to begin with? They contract Lyme disease, and then because of their underlying autoimmune issues or immunity issues or immune system issues, they can't properly rid their bodies of the spirochete by killing it, and instead they just get cloaked by the spirochete. And then that's why so many of us that are chronically ill have immune issues and Lyme disease, chronic Lyme disease as well.
Yeah, I think that a chronic infection scenario screams out that the immune system isn't fully revved up. And so it's like there are many ways that it may be partially crippled or totally incapacitated. And we don't need to go through all of those, but there are many opportunities for the immune system to be weakened or to be sort of put out of business. And you can, knock out the T cell side, you can knock out the B cell antibody side, you can knock out the complement side. There's many ways of, corrupting a healthy immune system.
So Dr. McDonald, before we move on to the next topic, I'd like to talk to you about Lyme disease and the number of Lyme disease cases increasing. And I recently read a book by General Stanley McChrystal, who defined risk formulaically as risk is threat times vulnerability. And we do know that the threat is increasing for a number of different reasons, but we're coming in more and more contact with ticks. But I'm also wondering whether or not the vulnerability piece of the risk analysis of the risk formula is also an issue, just sort of following up on the conversation you were just having with Matt, because it appears that we are more and more vulnerable to this disease in large part, or maybe in part, because of immune compromise resulting from a sort of living in this toxic soup. So can you talk to us about whether or not you believe that we are becoming more vulnerable as a result of sort of the lifestyle we've created for ourselves collectively, and whether or not that may be the reason why, or one of the important reasons why so many more people are being diagnosed with Lyme disease?
Well, we live on a planet that isn't sterile and it isn't pristine clean. And we do have issues of contamination. We have issues of new infections coming and going. We have our COVID, sad story with people who do well and people who don't do well. So actually, the vulnerability issue is very multifactorial. Depends on your lifestyle. I mean, I don't work out. Rich does, so he's going to be healthier than I am. So, all these things that work out in different ways are explanations for variability. I think that the number of ticks per unit area in various parts of the country seems to be increasing. It seems to be moving. We know it's definitely moving across into Canada. Canada used to have it as a rare disease. Now it's like an epidemic. And all the things that we had in the 80s on Long Island when we were living in tech heaven near Shelter Island, where the Lyme tick was first described. We were like less than 10 miles from Shelter Island. So we had like ground zero tick disease because we had a lot of ticks. And there are more species of the infection. So Lyme infections, and may have 30 or 40 different subspecies, and we're getting better at picking up the ones that are harder to diagnose. The ones that the Mayo Clinic picked up was a total new animal, wasn't it, out there in Minnesota? So there's new things coming along all the time. But health and wellness are very complicated, and the immune system depends on health for optimal functioning. But superimposed on that is that the background infection in the community for various things, not just Lyme, but viruses like COVID and other things, can also participate in having a bad outcome. And then toxicities and things that are in the water or in the air that we are exposed to and we don't really know about, but they do have an impact. So it's very, very complicated.
So on the threat front, you're arguing that, of course, there are more ticks, so we're more likely to come in contact with ticks, and we're more likely to come in contact with ticks on a regular basis. But you're also arguing that because the bacteria itself is changing, and we're finding more and more strains of this bacteria, that the threat piece of it is increasing. And then on the vulnerability piece, some of it is lifestyle, some of the lifestyle choices we are making as individuals. Some of it has to do with all of the different chemicals and EMFs and other things our bodies are coming in contact with, but also we're becoming more vulnerable because we're coming in contact with a more diversity of viruses and bacteria from other sources that are, of course, compromising our immune system. So it's really, yes, it's toxic soup, but not all of it our own making, or not directly our own making, because we're coming in contact with all kinds of other biological agents that are natural, that are also becoming immunocompromising, or causing us to becoming more immunocompromised and therefore more vulnerable to the threat.
Yeah, no, we're going to hit liver, we're going to hit chronic lymphocytic leukemia, we're going to hit glioblastoma, and we're going to hit suicide as horrendous situations for patients who have those. All of those in common have the double infection common link. So years ago, we had one, which was Lyme Borrelia burgdorferi to worry about. Now we have Lyme miyamotoi and Borrelia burgdorferi. And all of the next 4 cases are going to be examples of people who got double infected with miyamotoi and with burgdorferi. The double infection led to more severe and more significant disease than possibly a single infection might do.
All right, so let's pause there for a second, because one of the things that we quote you about all the time, and you probably don't know how much you influenced me in that, but we literally quote you probably every week on this podcast, if you didn't know that. And one of my favorite McDonaldisms, if I could use that term, is the divorce from the word Lyme, right? I mean, yes. You had argued very powerfully in an article you had written for LymeDisease.org, which is again, I think one of the most brilliant pieces you've written, quite frankly, is that we need to divorce ourselves from the word Lyme disease, because Lyme disease is essentially a disease without a definition, because it's so many different things. So you were one of the earliest people back in the 80s, that recognized that Lyme disease really is a polymicrobial disease that has a number of different permutations. And by using the sort of single term Lyme, we're putting ourselves into a box where we have a disease without a definition, right? So let's discuss that in a little bit more detail, because now you're starting to show the brilliance of your observation about the need to be divorced from the word Lyme disease, because the next 4 topics we're going to talk about are all really polymicrobial diseases, but in this case, you've been identified 2 different strains of the same bacteria, which when they come together create substantially more havoc in our body and make us more likely to become very sick than if we just had this one bacteria, right? So let's talk about the liver in this context, because I think you made a really powerful transition for us. Thank you for doing that. It makes my job easier. So the liver issue is a big one here in the Lyme community, and something we see all the time on our social media. We talk about all the time the importance of the liver and the role the liver plays in detoxification, and how, again, you just talked about how important that was in the brain, but of course everywhere. So let's first talk about the liver and what the function of the liver is, and then I'd like to take you to a second step and talk about how important it is to have a healthy liver so that we can go through the process of killing and detoxifying.
Yeah, the mission statement for the liver is that it purifies the blood. It regulates levels of important chemicals in the blood, like cholesterol and lipids. So it has a filtration function. It has an immune function. There's special immune cells in the liver, which are not found anywhere else in the body. And overall, liver health is very important to total body health. The early cases of doing, blood testing in Lyme had a bunch of patients with early Lyme disease who had liver functions that were whacked.
Okay, so now this is acute Lyme disease, right?
This was acute, yeah, this was acute. And it never got whacked enough that they got a liver biopsy, because it was up a little bit and then the enzymes were up there for a while and then it came down. But there was definitely a group of people who had very early disease, like when you had your erythema migrans skin lesion, that early. And at the time, blood testing would show some liver abnormalities, not major, but problems. So at that time, even then, the bug that got into the skin didn't stay in the skin. It was in the bloodstream, and it got to the liver and it got to the brain in some, considerable number of patients, and the liver healed itself. So that's important. Now, people who have chronic situations can have more chronic issues in various organs. Liver is one of them. Spleen is another. Brain is another. Eye is another. So chronic, it can set up in various sites, and they don't all have to be the same for everybody. So it gets very complicated. Chronic is complicated. But the other thing I want to pound into everybody's brain is that chronic equals biofilm. You can't have chronic without biofilm. Biofilm and chronic are as important as peanut butter and jelly. You can't have a chronic infection without having biofilms. They may be in the blood, they may be in tissue, they may be both. And biofilms are very, very resistant to antibiotic treatment, very stubborn. They're designed for survival of the bug inside the biofilm. And the biofilm complicates the biology of everything exponentially. And there's a great reluctance to deal with it cognitively. I mean, the Lyme community is sometimes not really biofilm aware, but biofilms and chronic disease go hand in hand. Does that so get to something there? Does that help you a little bit? Or
it's brilliant. It's absolutely. We're gonna spend some more time talking about it, but let's stay with the liver for another second, and then we can move on for that. So now, if you're a patient and you have acute symptoms, you have a bullseye rash.
Right.
Would you think it would be wise of a patient to demand that they have blood tests testing their liver function as a further step in the process of determining what appropriate treatment would be necessary?
Yeah, I think that would be a first step, because remember, you're treating the whole patient. You're not treating just the patient's skin and the red spot. You're treating the whole patient. And a lot of these liver function abnormalities, these low-level whacking of liver function, were kind of accidentally discovered. You had your blood test for your hemoglobin, and you got your regular chemistries, and you had your Lyme disease at the same time that those tests were being drawn, and you found a couple of liver things up, or maybe a few more, but not seriously. So yeah, and what that does is it also cements into the brain of the physician that it is from the start a systemic disease, that once the tick penetrates the skin and once it gets the spirochetes into the skin, then that is expressway, 12-lane expressway to all the other sites that provided by the blood. And where it may land up is unknown. Whether it lands up at any of the other sites is unknown, but at least has a chance at every part of your body.
Right. All right. So now let's talk about, you and Matt earlier were talking about how scary it is that this bacteria is so smart and it's so diabolical. Do you believe that the reason why we're seeing elevated levels in liver testing is because the bacteria is intelligently attacking the liver so that the detox function of the liver is impaired, and therefore the toxic levels of the blood increase and
toxic everything. I mean, it doesn't selectively pick out, oh, I'm going to just deal with this toxin or that toxin. If it's screwing up the filtration function, then everything is at risk, right?
And then the then the immune system is, of course, overwhelmed because there's so much toxicity, not just of that bacteria, but everything.
And the liver has its own immune system that's different from the system that's in the spleen. The different cells in the liver are different from the ones in the spleen. The spleen's another immune engine, right? And so the spleen can also and the spleen is also a filter, by the way. All the Babesia organisms in the world are taken out by the spleen, not the liver, right? So let's give the spleen a little applause too. So remember, we have a cooperative system, and we're not treating just a skin lesion or tick bite.
Dr. McDonald, I just want to point out again, I know you and Rich touched on this, but in this study done through Yale University, who you did some work for with this particular information and research you sent over to us, 19% of people with acute early Lyme disease had liver issues as a result of Lyme disease, correct?
That was early on.
Early on.
And then some of them become more chronic, more severe, but not everybody gets disease that's bad enough to get a liver biopsy.
Right.
You have to have really major chemistry and major symptoms in order to get a liver biopsy. So all of the things that are going on in the liver are underneath the radar because we don't biopsy the liver in healthy or mild liver abnormality patients. We don't biopsy it. So we don't know what's going on.
Understood. And I think a big part of this, though, is, as Richard said, that people that are having liver issues, when they start to treat Lyme disease, they become toxic, right? So this is, I think, a precursor. So Lyme is damaging the liver. Again, it's so smart. And then all of a sudden you start to kill off bacteria. You need your liver to function properly, and you become toxic, and it creates all these issues. But I do want to focus on, some of these terms here, right? Because we've heard about terms like hepatitis, liver infection, liver inflammation. I think one of them that you referenced is granulomatous hepatitis, right? Can you explain to us, what does this actually mean, that we're finding spirochetes in the liver in these granulomas, right? What are they? These are like balls of like blobs in the liver that are inflamed because of having spirochetes there? If you could give us some details to exactly what you're seeing in the biopsied livers, the ones that you were studying and the ones that were done through this research at Yale.
Okay, well, a granuloma is a collection of cells that may include the host white blood cells, the host macrophages, giant cells that are there to eat infection. So granulomas are the cells of the body trying to eat the infection, trying to contain it, trying to hold it in an area which becomes a round ball. So that's a cellular sort of a round thing, and it's a special kind of inflammation. It usually it always indicates an intracellular infection. So they can have infections where the bugs live outside the cells, and infection where the cells get infected on the internal side, and bugs are inside. And so granulomas are indicating intracellular infection. But so hepatitis that has some Lyme connection, we reported one case where the granuloma pattern was there in the patient, and the patient was treated and the patient did well. That patient, however, had been called quote, autoimmune patient. And because autoimmune patients and liver disease mean you get your name put on a transplant list, right, for liver transplant, because that's what autoimmune liver disease winds up with, you get, a slot on the transplant list. In fact, the patient had infection and had a disease which, although there was no other explanation, they said, well, it's got to be autoimmune then. Autoimmune liver, it's bad liver. We know what the disease is. Let's call it autoimmune. Let's put her on the transplant list. See how the sloppiness of thinking gets out of control?
But now what's wild about that is if that patient were to get a transplant, a new liver, it would have come back, right? Possibly because the Lyme was systemic.
Right. We don't have to go there either, but we don't want to unnecessarily transplant people for quote unquote autoimmune disease, which is actually infection. We don't want to do that. That's a no-no.
But the work that you're doing and the work that Yale's doing is finding that these liver issues, the liver inflammation, these nodules in the liver, right, the granulomas, et cetera, you're finding Lyme disease in the liver. And not only you finding Borrelia burgdorferi, but as you told Rich earlier, you're finding Borrelia burgdorferi and Borrelia, I think, miyamotoi in many cases as being the driving force together to create the liver issues. Is that correct?
No, these are creating severe liver issues, and that's a very small fraction. The total number of liver Lyme things that have been nailed down with research reports is under 10 in the world's literature. So there'll be people who will never see a Lyme liver patient in their entire practice because it's right now, fortunately, rare situation. And the infection interferes with a proper metabolic and chemical and physiological function of the liver so that the enzymes of injury related to liver injury go up in the blood. And they go up to a high enough level that they're called a high abnormal, result. That's Lyme hepatitis, infection causing chemical disturbances in the liver. And fortunately right now, it's a relatively rare condition, but it's a model for what's going on in people who have lesser degrees of inflammation and lesser degrees of infection who don't get up to that high level of abnormality in their liver testing results. So there's a spectrum. You can have a mild thing, you can have a moderate thing, you can have something that goes from abnormal back to normal because you recover, then it can regress or return, it can relapse. And Lyme is fundamentally a relapsing infection. We know that, you get better and then you have a relapse or get reinfected. So that's one of the liver issues. Liver is an important organ for health.
Okay, so obviously this is a rare disease as far as being diagnosed. But so far, but as you had indicated, Dr. McDonald, many people may not have the levels high enough when they were tested at that time because this is a relapsing disease, right? And also, I can't help but wonder how many people have elevated liver levels when they get their blood work done and have it dismissed or misdiagnosed or misattributed to something else when really it's connected to Lyme.
Yeah, I think that number is a significant number, but if it doesn't get up to a very high abnormality, a high number in the blood of abnormality, they won't biopsy the liver and find out what's going on. If you don't biopsy the liver, you'll never know what's causing the liver injury. It's like in church, you take a collection and you do a collection plate, right? In diagnosis, you do a biopsy to find out what's going on. You want to find out what's going on in an organ which is disturbed, you do a biopsy of the organ and they have a better chance of figuring out what's going on.
And many people with liver issues don't get a liver biopsy unless they're severe cases, like you just said, with high levels.
That's right.
Correct?
Absolutely.
So again, it just makes me wonder in this discussion, because we know how systemic and debilitating Lyme can be, how many people have an underlying Lyme disease and, different strains, Borrelia miyamotoi, Borrelia burgdorferi, contributing, and they're just waxing and waning their whole lives because they're getting a little bit better, getting a little bit worse, and they never get to the root cause of what's going on. So I think this is another topic, Dr. McDonald, that down the road, we're going to be realizing, Dr. McDonald warned us about this, and here, we're starting to learn more about the real things that are going on. So is this a reason why somebody gets bit by a tick, right? And they maybe are mildly symptomatic or they're a sensitive person, that the use of antibiotics is really an important thing at the acute level, because you don't want to get it systemic into your liver. You don't want it to get systemic into your tissue. And by treating at the acute level after a tick bite is probably worth it, even though there may be some negative side effects compared to what the damage can be if you get disseminated Lyme disease.
Yeah, here's a little sidebar. There was an antibiotic that was popular when my wife had her treatment with Joe Burriscano. It was called Ketec. And Ketec, early out of the box, was looking very good for treating people who have chronic Lyme disease. And over time, it was taken off the market because it was considered dangerous in Lyme disease. Why was it dangerous? Because the liver got whacked.
Mm.
Now, can we do the math? Can we connect the dots? Maybe the liver got whacked because there was disease in the liver that needed to be dealt with, and the Ketec was dealing with it, and the Ketec wasn't fundamentally toxic, but doing its job, and therefore the enzymes which had to go up and go down, represented occult Lyme in liver, occult Lyme hepatitis, which was perceived to be quote unquote toxic Ketec. Remember? We've had toxic amyloid, right?
Yep.
We've had toxic alpha-synuclein, right?
And now we're realizing that those were
maybe,
incorrect.
maybe not. But I mean, going back and learning with 2020 hindsight, if we didn't do the liver biopsy, we wouldn't know what's going on. Right now, the only thing that they do is look what's in the blood and say what the enzymes are and say, oh, well, there's trouble in the liver. And we don't feel brave enough to biopsy the liver and find out what's going on. I think there's a point now where you're going to have to be ready to biopsy the liver in people who have persistent abnormal liver enzymes to see what's going on. Is it Lyme in the liver? Do we have more than 10 cases on planet Earth?
Oh, no doubt.
Of course we do. But how many have gotten through the publication and peer review and all that? How many have actually had a biopsy, right? Very, 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 very small. And the liver is an organ of health. You've got to have a healthy liver to recover. So let's get some biopsies going here.
So I want to jump from the acute to the chronic, right?
Yeah,
because in the literature you sent over from Yale that you worked with, there was a case of hepatitis associated with chronic Borrelia burgdorferi infection. And, this is a case report from 2014. And essentially, I believe what this is saying is that there was hepatitis or liver inflammation that was related to a Lyme infection. But after a course of antibiotics, the Lyme bacteria persisted and the liver inflammation persisted as well. Is that correct? Am I interpreting this study correctly?
Yeah, I know I sent you the studies, but I don't have. I sent you maybe 6 or 7 studies, so I'm not exactly sure. But I would defer to what's written and not to my memory about the details, because I've been, doing a lot of other things, and so I can't tell you for sure the details of that one case.
I can just read you the quick snippet. It says, we present the case of a 53-year-old woman diagnosed with a Lyme diagnosis who developed acute hepatitis with elevated liver enzymes while on antibiotic treatment. Histological examination of the liver biopsy tissue revealed spirochetes dispersed throughout the hepatic parenchyma, I'm probably mispronouncing that.
Parenchyma, that's right.
Parenchyma, and the spirochetes were identified as Borrelia burgdorferi by molecular testing with specific DNA probes. Modal spirochetes were also isolated from the patient's blood culture. So not only the patient's liver, but in their blood. Yeah. And the isolate was identified as Borrelia burgdorferi sensu stricto by 2 independent laboratories using molecular technology. So 2 independent labs diagnosed Lyme at the blood, and the liver biopsy confirmed Lyme in the liver. These findings indicated that the patient had active systemic Borrelia burgdorferi infection and consequent Lyme hepatitis despite antibiotic therapy, is what this case study showed. But I think that's powerful. I think that's an important study to focus on. So can you talk a little bit about that, Dr. McDonald?
Well, that may be the case where they had a nice picture, a yellow picture of the spirochetes in the liver. They had It had not just a few spirochetes, it had wall-to-wall spirochetes. You look at the picture and there's no spaces between the spirochetes. I mean, they're all almost shaking hands with each other, they're so close. So yes, that was a situation where they had over the top done the molecular proof to say, yes, it is absolutely thus the Burgdorferi strain, and it is, there's no doubt that it's some other spirochete. One of the other spirochetes that can cause trouble is the leptospirosis, infection, and that one can do terrible things to the liver too. So you can have acute yellow atrophy with leptospirosis and fatalities in a condition called Weil's disease. It's a leptospirosis in the liver. Syphilis could get into the liver and cause terrible things to happen too. So it's not just Lyme disease as the bad kid on the block. All spirochetes can do terrible things in the liver. And you have to be aware of it, and you have to firm it up with a biopsy, because you can't, with blood testing, tell whether the spirochetes are just in the blood or in the blood and the liver. You can't tell. And if it's in the liver, you want to treat them appropriately, because when it's in the liver, it's there big time. It's not there one spirochete here, one spirochete there. It's big time. And it's a perfect thing because the spirochetes need, fatty things, and the liver is the gatekeeper for fatty things to keep things going. So liver extract has actually been used in some culture medium to enhance the reliability of growing spirochetes from blood in some cases. So, liver is important.
But I think this study also proves that it persists, right? Because there's this term out there that Lyme persists, right? Persistent Lyme or chronic Lyme disease. Hey, I treat with antibiotics, but I'm still sick, which we know to be true. But this study out of Yale that you've worked on is definitively proving not only that chronic Lyme exists in the blood, but with 2 independent labs, but also chronic Lyme exists in the liver, correct?
Yes, big time. When it gets to be, ensconced, entrenched, dug in, right, in the liver, you're not going to have one here, one there. You're going to have many spirochetes in the cases that we've seen so far. It's a high infectious burden in liver situation.
And not to get too technical with the forms, but this has always fascinated Rich and I. And in all of your photos and in your research studies with the liver biopsies and even the blood lymphocytes, which we're going to get to next, you talk about different forms of the Lyme bacteria that you're finding in you're in the laboratory environment and using your fish testing. So you talk about the whole spirochete having a granular form, a cystic form, a biofilm form, and I believe also like a straightened form. And, can you just explain to us what these different forms are? Because, we've heard that the Lyme disease bacteria can, shapeshift, where it can evade the immune system and evade the antibodies by going from a spirochete to, something benign. But if you can just give us, some high-level overviews of the various forms Lyme disease can take and which forms you believe are probably the most dangerous to make us sick.
Okay, well, I was the first to put the cystic form of Lyme disease on the map. I did that by discovering a cystic form in an Alzheimer brain. And so the cystic form is round, and it is sort of like a mechanics ruler where, you have an area where the ruler is coiled up, and then you pull the ruler out and you can see the measurement, and then you roll it back into the container, and the spirochete, not the spirochete, the ruler is coiled up inside it. It's kind of like that. Where
like a measuring tape?
The measuring tape, right. Okay. Yeah, like a mechanics or carpenter's tape. So when the spirochete is in a hostile environment, it will go round on you. Sometimes and become a round body. And then after it's returned to a healthy environment, it can uncoil and become spiral again. Some spirochetes are not coiled, but they're still real, and they're not coiled because they lack the flagellin. They've got a mutation, but they're still capable of causing trouble. Some spirochetes actually break up into little pieces, so that if you take and mince the spirochete along its long axis and make these tiny little pieces of mincemeat out of the spirochete, each one of those has a chromosome, and the whole chromosome copy number in spirochetes can go from 16 whole chromosomes to 50 in long spirochetes. Each chromosome can reproduce an entire long spirochete.
So a fragment of the spirochete can regenerate into a full spirochete.
Yes, and so it's like a biological multiplier, because if you kill off the cylinder and you release chromosomes, 16 copies, then you'll get 16 regenerations of spirochetes. So it's a biological multiplier.
So you kill one spirochete thinking you're doing good with antibiotics, and then it creates 16.
It goes granular on you, right? Or it goes cystic on you and granular. And then all of those can continue. So it's a way of getting past killing, because you can't kill the granules.
So I'm
The
sorry,
only way-
the
yeah.
cystic is if the spirochete's in a hostile environment, it'll stay alive, it'll go into the cystic or round form and then go back into a spirochete when it's safe again for the spirochete. But the granular is when the spirochete breaks apart and the spirochete breaks down into little mini pieces, and then each mini piece can essentially create its own spirochete again.
Mincemeat, right?
Is that's different than remnants, because remnants of Lyme disease is generally dead, right? Or am I mistaken?
That's right. Yeah, those are dead, but the granules are actually living.
Wow.
So that's scary too, because they are they have the whole chromosome. They've got a wrapper around it that protects it, and so the granules are there to regenerate. And the granules can sprout little tails, and then the tail gets longer and longer. It becomes a spiral thing again. So yeah, that's another life form. So there's many different ways it can survive assault and persist. We could do 2 hours on just the morphological differences, because I haven't gotten into cell wall deficient forms or the other forms that are out there, the ring forms and other things. So I don't want to carpet bomb your listeners with too much data.
Well, while we're still on the liver topic, I mean, obviously you were brought in to consult with Yale, and they had a ton of supporting studies. So I know there was, I think, like 9 or 10 studies that you provided us with as backup in addition to the work that you did. So is there any traction there? I mean, are you doing anything moving forward with the study of livers and Lyme disease? Is Yale pursuing anything? Are you aware of anything going on out there in the world that's going to help advance what you've already done in this arena?
The Yale School of Medicine does not have a single faculty member who is studying Lyme disease in the liver as a focus of research. That's ironic, because Yale put Lyme disease on the map with Alan Steer at Yale. Alan Steer went to Mass General, and his pathologist, Paul DeRay, left Yale and went to other places. So after those two people left, there was no interest in Lyme liver pathology at Yale. To this day, there is no interest in Lyme liver pathology at Yale. That paper that came from the past was when they had somebody there who was interested, but that person is no longer there.
All right, Dr. McDonald, let's take the next step and transition over to leukemia.
Oh boy, here we go.
Yeah, this is something that is regularly discussed in the Lyme community, and I think it's important that you've given us some insight here. And of course, we do want to give everyone the caveat that this is not something that's going to happen to you. This is not something that's necessarily going to happen to you. This is just something that we're now seeing in this disease that has been defined as the great imitator. This is another form of imitation, and in many cases, people are being diagnosed with with leukemia and a form of cancer that either is triggered by Lyme or is really just Lyme shape-shifting and creating some other issues. So why don't you talk with us about your findings on CLL?
All right, well, the lymphocyte is part of the immune response, and a healthy lymphocyte, when exposed to live Borrelia bacteria, will divide and go into what they call a blastic transformation, and the number of lymphocytes will get increase in number, and the lymphocytes will get bigger in size, and it will be switched on metabolically. And you have the T cell lymphocytes, or the cellular immune part, and then you have the B cell lymphocytes. And all of those things are then stimulated by exposure to live Borrelia spirochetes. Now, as of this point, there have been peer-reviewed papers published which describe in Italy and in Europe lymphomas, which are actually lymphocyte proliferations inside of lymph nodes or solid tissue. Lymphomas are not leukemia, but lymphomas may have a leukemic phase.
Is lymphoma cancer, Dr. McDonald? I just want to make sure. Okay.
It's a blood cancer. It's a cancer where the lymph nodes get enlarged, and the lymphocytes cause tumor-like accumulations in skin and other organs. So they found in Italy that there's a good connection, a durable and robust connection between people who have certain lymphocytic lymphomas, non-Hodgkin's lymphomas, and Lyme infection in Italy. And so they've drawn a connection between those two things. There are also papers which describe people who have chronic lymphocytic leukemia in this country and in Europe, who also have Lyme disease infection, and they can't decide which came first, the chicken or the egg, and whether the leukemia would be there as an incidental rather than a related thing, because the immune system in leukemic patients is going to be suppressed. And so you get an infection in a suppressed patient, just like you get an infection in an HIV patient, Lyme infection in a steroid patient, a Lyme infection in any other cancer patient. So just because you have the two things going on in the same body doesn't mean that they're connected by cause and effect, right? But the certain types of leukemia will sprout out of solid lymphoma cases. So you can have a lymphoma that starts out as a solid tissue thing and then evolves so that the cells get into the blood and they have a leukemic phase. And chronic lymphocytic leukemia is one of those categories where you have, as the stages of the disease develop, you can have gradual small increase in the number of lymphocytes in the blood that doesn't reach the threshold for calling it chronic lymphocytic leukemia. And then over time, it gets a higher and higher number, and eventually it crosses the threshold and becomes numerically sufficient, high enough count in the blood to say, okay, 50,000 lymphocytes in the blood is leukemic, 10,000 lymphocytes in the blood is not leukemic. So you have to reach a certain number threshold. I was lucky to receive blood from a patient who 6 years, in 2016, wanted to have me run my tests on her blood. He said, oh, I like what you researched. I'm going to send you some of my blood. So I got it, I put it away, and then I came back to it recently, and I looked in it. I found that the blood is loaded with spirochetes. And I said, ooh, well, that's very, very interesting. And what else is going on? She said, well, back in 2016, I was okay, but about 3 years ago, I was diagnosed with chronic lymphocytic leukemia. And it happened that I had lymph nodes in my armpit, and there's a breast cancer risk in my family. So I figured, oh, I'll get it biopsied, see if it's breast cancer, God forbid. And they biopsied it, and they found out that it was a lymphocytic lymphoma, chronic lymphocytic leukemia. And then so she was then diagnosed 3 years into the 2019 as chronic lymphocytic leukemia. And she's doing okay. She's smoldering along. She's not in any kind of danger. And I hope that she'll do what many patients do with chronic lymphocytic leukemia and go for 10 years with no problem. It's just have a high lymphocyte count in the blood. But because it was Lyme disease in her blood, and now it evolved from no leukemia to lymphocytic leukemia, and because the Italians had described Lyme disease causing lymphocytic lymphoma, I said, I'm going to take a whack at this and see if there's anything I can find in the blood that connects Lyme disease spirochetes with her chronic lymphocytic leukemia cells. And I hit pay dirt. Oh my, did I hit pay dirt. And so with my DNA probes and my other testing, I found that the cells in her blood are that contain inside the cell Lyme disease infection, Lyme disease inside the leukemic cells. That's a first in the world. And that's going to get me into big trouble with the leukemia and lymphoma people too, because that I wasn't supposed to find that, but I did find it. So that's where we are with that case. I've sent you pictures, and you have the handout, which you can provide the listeners, which will show the people what the Lyme disease spirochetes look like as they wrap around the lymphocyte and surround it like a bracelet or like a corset. And then they work their way inside the lymphocyte, and they get inside the nucleus, and then you can see the spirochetes inside the nucleus of leukemic cells. So it's doing the same thing it does with other places. It gets a foothold, and then it wheedles its way into the inner part of the cell, and then it wheedles in its way into the nucleus. And that's a first in the world. And it's pretty straightforward. There's nothing, wishy-washy about this. This is like dramatic. This is dramatic.
So I want to just kind of break this down, right? So the Europeans in the past have identified a connection between Lyme disease and skin cancer, because that, I think you call this
Lymphoma in the skin.
lymphoma, right? Which is really essentially skin cancer, correct or no?
It's actually a lymphoma on the skin, just to make sure that we focus on the lymphocytes rather than the epidermis and the red spot. You know, you can have lymphoma of the skin and have some red, but it won't look like erythema migrans. It's usually an area which is a bump. It's just all a piling up of lymphocytes in the skin, which is like a tumor.
And the
So it's a
lymphocytes
tumor
are,
like
are
that.
white blood cells. So it's
They're
white.
lymphocytes. Yeah, they're not. They're, all cells in the blood could be called, if they have a nucleus, could be called white blood cells. You have the neutrophils; those are the acute inflammation. These are the immune cells. These are lymphocytes. They're small, round lymphocytes with skimpy amounts of cytoplasm. They're the immune cells in the blood.
Gotcha. So the immune cells are the ones that glob up and form the lump on the skin with the Europeans, correct?
Yes.
Okay. So this was quite, and now was this dangerous? Was this just an inconvenience with like a lump on the skin? Is this cancerous? What's the health risk that these people experience with the European model and the European studies?
It is by definition, because they call it lymphoma of the skin, it's a cancer of the lymphocytes in the skin. Now, in Europe, there's a fascinating condition called Borrelia lymphocytoma, which is related to Lyme disease infection in the skin, where you get bumps in the skin and you get lots and lots of lymphocytes in the skin, and they have, you know, lymphocyte proliferation in the skin, but it's benign. It's just related to the infection. So you could look at it as the first stage between infection to lymphocyte immune system, making a lump in the skin, which is benign, to lymphocytes in the skin, which now become cancerous. So it's a beautiful progression. Borrelia lymphocytoma to Borrelia lymphoma in Italy in the skin. It's like, ooh, and that's all due to that spirochete in the skin. It's stimulating the cellular side of the immune response, not the antibody side so much, but the cellular side. The lymphocytes themselves are dividing, and getting active, and they're making a lump, and that's a tumor, and the tumor can be benign, Borrelia lymphocytoma, or it can progress to become a malignant situation. And when it gets into a stage where it starts releasing the cells into the blood, it becomes leukemic lymphoma. You can have lymphoma without leukemia, or lymphoma with leukemic release of cells into blood, and that's how you get to chronic lymphocytic leukemia.
So this is truly diabolic. Again, this is something
It's diabolical. Yes,
is
it
just
is
insane.
diabolical.
That,
Yes.
So wow. So it's really a progressive disease, and it can start off and then progress all the way through to leukemia and aggressive cancer, depending on how far it goes in that case. Now, what you did is slightly different, because that was what the European studies did. You were studying something different. This chronic lymphocytic leukemia that you were studying, talk to us about how that was different than the European studies. I want to make sure we understand how you built upon that, but in a different way based on the European research.
Well, I talk about, infection in skin to benign lump to malignant lump, and then the lump in the skin, which is the cancer, releases cells into the blood and becomes leukemic, right?
So you brought the final step. You found the final piece of the puzzle.
I just jumped to the final step and say, okay, I'll work backwards. Let me go backwards from leukemic cells in blood of lymphocyte immune type and work backwards to the infection.
Understood.
Instead of going forward, I work backwards.
So you're the one who made the chronic lymphocytic leukemia, the cancer, the everything being released out into the blood. That's the connection you made from the beginning with the European started, but you finished the work the Europeans couldn't finish, basically.
Well, I don't know. Somebody's going to finish me off for all this. I'm going to be found in a dumpster somewhere. Yeah,
I'll say it. You don't have to say it. You finished off their work and you brought it full circle. I mean, just to show how severe this disease can be. So, and this was coincidental. A patient that you were just working with had crazy spirochetes, and then you found this CLL connection, and then you were able to work backwards and identify that and this is the crazy part. And again, I want to tell our listeners that we're going to put the reference documents. You put together this beautiful presentation, Dr. McDonald. Thank you for that. We're going to link to that in our show notes, so our listeners can go and open that link in our show notes of this podcast and view everything you put together for them, because that really is extremely powerful to see the spirochetes invading the lymphocytes, right? And how that process happens. And if they invade the lymphocytes, that's the beginning of the end, essentially. You discovered the end piece, is what we're saying here, right?
Yeah, I mean, it's the DNA probes have come to my rescue so many times. And without my DNA probes, I would not have anything to talk about. But with the DNA probes that I have for both Miyamotoi and for Bergdorferi, and remember this leukemic patient has double header. She's got Miyamotoi and Bergdorferi. It sets her apart from Lyme patients, Borrelia patients who only have one type of infection. So let's not get people jumping out of windows because they've got Lyme disease and they're going to worry about leukemia. You may have a double infection in another set of circumstances which move you from reactive, benign to leukemia. I don't want everybody to get, crazy about this.
Not everybody will get to the end result. It's a certain set of circumstances. Understood.
Not everybody gets everything, right?
And also, if you start to treat your condition now, you can prevent it from escalating as well, correct? So if you start to address it and treat, you can prevent the evolution or progression of whatever's going on in your body, right?
Exactly. That's like early diagnosis, early treatment, good result.
And I think the same is true of everything we're discussing, even when it comes to the Lewy body dementia. And correct me if I'm wrong, but if somebody were to get accurate treatment and to start to address the spirochetes and address, the biofilm and address, everything in their brain, it can stop the progression of the dementia, but it can't reverse the nerve death that you described, right?
And remember, normal people who don't have infection will lose a certain number of nerve cells every day, particularly if they go on a bender, right? So, loss of nerve cells is part of life. But with the accelerated extra part of the infection, then it puts you at a disadvantage because in addition to what you're normally going to lose, the infection adds additional nerve cell loss. And that puts you into, gray zone.
And I think though, so Borrelia miyamotoi is becoming more and more common. And I think, you said earlier, Borrelia miyamotoi is coming into its own, meaning it's starting to evolve, it's starting to grow.
It is.
So unfortunately, these cases are going to become more and more prominent over time. And they're probably, much larger today than we think they are because we're not doing proper tests or studies. And nobody really has any of this information that you're sharing with us today on this podcast. So I think that's an important note too, to share. So with this combination, right, of Miyamotoi and Bergdorferi, why do you think that drives it so much harder and makes it so much more likely that you're going to progress to get the full-on cancer that you described? And, in many cases, if you just have Bergdorferi, maybe it won't progress that badly in that case.
That's $1 million question. I don't know. I guess right now it's an observation. That's all I can say. But I'm seeing it in different settings clinically. I'm seeing it in the liver. I'm seeing it in the chronic lymphocytic leukemia. I'll be seeing it in glioblastomas. And, all of these things are significant threats to health that have occurred in people with double infection. That's all I can say. I don't want people to jump out of windows if they have, Lyme disease and say, oh, I'm going to get glioblastoma. No, you're not going to get leukemia. And, not everybody's going to get everything. But the people who do have the bad disease, we can understand that there is a role for double infection in their disease getting a foothold in their body.
And this work, this blood lymphocyte work that you just did, the CLL, I think you just submitted a paper or at least a cover sheet on this just this past month, August 11th of 2022, right? So this is something that you've been recently working on, and it sounds like there's going to be some more traction, hopefully, in the near future with this as well.
Yeah, I have a good friend who's director of a chronic lymphocytic leukemia program, and his professorship is just in chronic lymphocytic leukemia, and he practices in Long Island, which is Lyme, central.
Right where we are.
So I'm going to be looking out to get some more specimens from Long Island CLL patients and see what I can find in those patients. And I hope that I can find some interesting results with that.
All right, Dr. McDonald, let's now pivot over to something we talked with you about last time, which was the work you're doing on glioblastoma. And it's important to talk about this because this is a form of brain cancer that seems to be accelerating. We know Senator John McCain died from glioblastoma, and there was a finding of spirochetes in his brain autopsy. We know the president's son died from glioblastoma, and I did share with you during the last podcast that I have a brother-in-law who died from multiform glioblastoma after he moved to Long Island after he married my sister. So this is becoming more and more prominent in the news. It's becoming a part of our lives, and certainly many elected officials and their families are being touched by this. So let's first talk about what your findings were what your findings were when you did the research and we talked last time, and then you made some new findings between our last podcast and now, where you've learned there were some open questions last time that you've now found answers to.
Yeah, I think that the paper that I published in 2016 had cases, 2 patients, and I used my DNA probes. I had little pieces of the glioblastoma tumor. They drilled a hole in the skull, and then they went in and they took little pieces from the center of the tumor, and they sent those little pieces to me, and I did the DNA probes, and the probes for Borrelia burgdorferi lit up like a Christmas tree. I mean, there was so much signal there. You could read the New York Times by the amount of light that was coming out of the fluorescence of those tumors. And it was so bright that you couldn't tell whether it was outside the tumor cells, between the tumor cells, in the tumor cells. You know, you couldn't see where the tumor cells stopped and the blood vessels and other things continued. So all I could say was that there was a strong signal and a DNA signal. Then I got the idea that with my new methods of immunohistochemistry, that I could do a mapping of the individual spirochetes without having fluorescence, but having brown or black color of the spirochetes in the area of concern. And with those different techniques, I could now cone down an individual glioblastoma cells and see that inside the cell, there are black spirochetes, or inside the cell, there are brown spirochetes. And so I sent you pictures of that in the handout. And so that's the update, is that the spirochetes are inside the tumor cells in big number, or wrapped around the tumor cells like a corset. And also between tumor cells, there are blood vessels, and those blood vessels are part of the tumor. They're growing crazy, and in those blood vessels, there's spirochetes. So there's spirochetes all over the infield and outfield in glioblastoma. And that's the new update there. It shows that it is inside the nucleus, therefore it's driving the malignancy from a DNA point of view.
So one of the themes that we seem to be developing here with all of the issues that we've talked about so far is that once the bacteria invades the nucleus, that's when we're really in trouble, right? There's really no coming back from that, and that's why we have to make sure there are a number of different steps, and you went over the steps between the tick biting you and ultimately, the bacteria getting into your bloodstream and then getting to you, then getting past the blood-brain barrier and getting into your brain and then getting to the cells. And I mean, there are just so many different steps before you ultimately have this fatal event for your
worst case, let's say worst case scenario in the event. Let's not
no, but I'm saying I'm just talking about the cell.
Glioblastoma is always fatal, you're right, but I mean,
Yeah.
In other situations it's a hepatitis, which isn't fatal necessarily.
But
CLL is not going to be fatal. Yeah.
I don't mean for the person fatal, but it's fatal for that cell, right? I mean,
the cell is labeled for death. Right.
Yeah, once it invades the nucleus, the cell itself is no longer viable. And you did share with us that losing some brain cells is not a unique opportunity. You can go out and get liquored up, and you're gonna lose some brain cells. So it's not it's not going to be fatal to us, and there is coming back from that. But of course, it is always important that we work back, not only because we want to inspire people to grit through so many of the challenges they have to grit through in order to be able to deal with this, right? But it's also important to know that when people in our families are sick, in many cases there is a solution that isn't necessarily so obvious at first glance, and in many cases, there is a tie to Lyme disease, and it should also inspire all of us collectively. Putting aside just the folks who are largely followers of this podcast, but it should inspire folks collectively to make sure that they're protecting themselves at the earliest stages, that they're avoiding coming in contact with ticks, that they're checking themselves for ticks, that they're getting early intervention, that you've also given us some new tools. For example, let's bounce back for a second to the liver studies. I certainly don't think, when you're taking antibiotics prophylactically, that you should stop taking the antibiotics until your liver levels are stabilized, right? I mean, that should be a test that our primary care physicians are using when determining how long we should be using antibiotics, and not falling back on some of these, these general, guidelines. I shared with you that when a couple of years ago, before we started this podcast, when I was bitten by a tick, I went to my doctor's office and they gave me 5 days of doxycycline, and that was it. You're better, right? Well, there was no testing. There was no follow-up testing other than 6 weeks later testing me for the Lyme bacteria only, and I was bitten by a Lone Star tick, which doesn't harbor, the Lyme bacteria, right? So, I mean, we have these sort of like these very general, guidelines that clinicians are using, and they should be using patient-specific testing. And I would argue, based on the conversation we were having, that some of that patient-specific testing needs to be liver testing, liver level testing. Even if you're not going to the biopsy level, you have to make sure that liver function is normalized before you stop the doxy.
I couldn't agree more. I think that's crystal clear.
All right, so let's move on to our final topic for the day.
This is. Are you tired yet? Are you getting?
Listen, we would keep you forever. You can't wear us out, but we'll wear you out if you want to spend this any longer. Because we also have a request that we're going to make of the community after we talked about this last topic. But of course, one of the most painful and most scary topics in the Lyme disease community is suicide, and we know that many people in the Lyme disease community are dealing with various neurological and emotional mental health issues, and in many cases, folks think that Lyme disease is a choice that's being made based on the emotional issues or the mental health issues, or just the absolute exhaustion that they're suffering from. But I think you actually have something new to add to this based on the research that you're doing. So can you share with us what your research is on the blood tissue? I'm sorry, the brain tissue that you had received from someone who had committed suicide, and what your findings were specific to that research.
Okay, this goes back to 2016. I received a request from a physician who had read about my work. His daughter at that time was being evaluated for Lyme disease testing, and he thought that it might be that she had Lyme disease, but the infectious disease people could not confirm it. So he sent blood from her, and I was going to do DNA testing. I did DNA testing, and I photographed the spirochetes in her blood, and they were there in high number. Biofilms in her blood, they were there in high number. And I wrote back and I said, I have DNA evidence and pictures of the spirochetes in her blood. So she, whether or not the antibodies are helpful or not to you, these pictures should be helpful to you because you can actually see the bugs in her blood. They're there in big number. And I'm using DNA probes, so there's no false antibody considerations here. It's all DNA stuff. And, the pictures I sent were pretty good textbook pictures of what the spirochete in the blood should look like. And he shared my report with his infectious disease consultant, and the consultant said, I don't really think this is credible work. Her antibody tests are negative, therefore I'm not going to treat her. And so she didn't get treated for the infection. Over the next 6 years, she had spiraling down with psychiatric disturbances, multiple hospitalizations, psychosis, just deterioration and institutionalization. And she came in and out of the institution, and she tried to commit suicide 15 times. Not once, but 15 times. And the 16th time she succeeded. And they decided to have an autopsy and have the brain from the autopsy, submitted to Columbia University, which has Lyme disease brain study. And Columbia got the brain, and they proceeded to try to find evidence of spirochetes in a brain. And they worked on that for January to October, and they were unable to conclusively find any spirochetes in her brain. And I said, well, I know about the case. The father had contacted me. I know that the brain came to you. I would really like to have just a small amount of tissue. Would you send me 20 slides from the brain, and I will see if I can find it with my DNA probes, because Columbia doesn't have my DNA probes. And so they were kind enough to send it to me. Remember, they've been looking at it for eight months and couldn't find anything with their basic stains for spirochetes. And I got the slides. I processed it for the DNA probes, and 2 hours later, I found that she had wall-to-wall spirochetes in her autopsy brain. Not subtle. Only with the DNA probes. The regular stains didn't show it. So that was it. Now, I've shared the results with the family, and the father was having a lot of trouble coming to terms with the fact that I had been right and I had been shouted down in 2016, and his daughter didn't get treated. All of that, he came into a certain amount of distress about that. So that's where we were. Now, after I found spirochetes in the brain with my test, I did my DNA probes on one slide. And in 2 hours, I found wall-to-wall spirochetes in one slide. That's not the whole brain, just one slide from the deep brain nuclei. And I turned that around, and the next day I told Columbia, this is what I found. I sent them pictures. I said, oh my God, these are beautiful pictures. And that was it. I was thinking of reporting the case, but I have held back because it's Columbia's case. And so, the only way I could actually report it is to put this together with the 6-year-old blood studies, which showed she was loaded with spirochetes 6 years before she died. And then all the years leading up to her suicide, that's 6 years of 15 suicide attempts, and then the death, and then the brain. I think eventually, since Columbia may not publish it at all, I'll publish it in some form. I was thinking of putting it in JAMA Psychiatry so that, people can look at it. Columbia has done studies with the Danish Lyme community, and they've found an excess of suicide risk and suicide activity in Danish patients who have chronic Lyme disease. So that's out there. That's not controversial. The icing on the cake was that I went into extra studies with my DNA probes, and I went to find evidence that the spirochete had worked its way into the neuron, just like I found in hepatitis, and just like I found in chronic lymphocytic leukemia. And no surprise, she has brain neurons that have been skewered like a shish kebab. The spirochete has gone into the neuron, and it's in the nucleus. And I've got many, many pictures of spirochetes inside the brain cells labeled for death inside her suicide brain. And you have pictures of that in the handout. You'll see them, and I'll actually enhance the handout so that I'll put arrows on it in the new version, so that when you actually share it with the group that listens, they can see just the way you can see where the spirochetes are.
Well,
that's it.
Thank you for sharing that story. I mean, look, the question I want to follow up with, I have several, is when you have chronic Lyme disease, putting all the physiological stuff aside, and the spirochetes invading your brain, and the spirochetes invading your neurons and causing the psychological conditions, you can't help but be sad and depressed because your life is so significantly altered. So I know many of us in the chronic Lyme community often wonder, is it really the Lyme bacteria causing me to feel this way? Or is it just how I am because my life is so different now? So what is your view on that, Dr. McDonald? Are you telling everybody listening to this podcast that yes, there's a piece of it that is you are sad and depressed because your life is now different, but also far beyond that, there is a component of the spirochetes causing psychological illness that you wouldn't have had otherwise?
Well, in the cases that we've discovered here with liver, with leukemia, with glioblastoma multiforme, and with the suicide patient, the common feature is that all of these severe or life-threatening or life-ending scenarios in chronic Lyme disease can be wrapped in the same package as spirochetes getting into the nerve cell, and then causing trouble with the nerve cell, or eventually maybe killing the nerve cell. So if the nerve cells are killed, you're going to be in a situation where it's a severe illness, and what can I say? It's just these are severe. These are at the extremes of what can happen in some people, but not everybody who has Lyme borreliosis. But some people who have the worst case scenario can get this because it's gotten into the nerve. So the goal is to keep it so it doesn't get into the nerve and doesn't get into the nucleus. It doesn't kill the cell.
If it's in the nerve,
it's outside. If you can kill it outside the cell, you're better off.
But if it's in the nerve and in the nucleus and inside the cell, that can cause psychiatric symptoms that you wouldn't have otherwise have had. Is that correct?
Yeah, I mean, I don't know how loss of nerves or inflammation actually causes a psychiatric product, but there are many psychiatric illnesses, and not everybody has the same study. If your brain is studied after you pass away, they don't all show the same things under the microscope. So it's very, very, very complicated. It used to be said, and I still think it's true, that man is incapable of looking at his own brain or his neighbor's brain and figuring out how the brain works. It's above my pay grade. I can't do it. All I can do is saying, I'm going to ring the alarm, send the alarm bell out that there is a disease process that has gotten into the nerve or gotten into the brain, and that's all I can tell you. I can't tell you how it's going to play out downstream, whether you're going to get better, whether you're going to have a chronic problem, a disability, an illness, God forbid, a tumor. I can't tell you which way people are going to go with this different pathway. But the common feature is that the spirochete invades the skin, it invades the blood, it invades the lymphatic system, it invades different organ systems. It sets up housekeeping in various sites, and not everybody gets every site the same way. It can set up in the eye, it can set up in the, there's so many possible combinations. It's just like the biology of syphilis. Everything that I've learned in my research, I have stolen from syphilis. I've stolen it. And I followed the model that was given to me in medical school. This is what syphilis can do to the body. I read the textbook, I committed it to memory. I just said, okay, I'm going to use that model as a model for my research. And I've used it religiously, and it's paid off for me. Paid off. And, not that I've gotten money from doing any of these things or, charge anybody for any of these tests, but intellectually it's paid off for me. I want to give back, because I can't write prescriptions for people, so I can't give back that free medical care, but maybe I can give a little insight to people so that they can understand their situation a little better. That's all I can hope for. This is my way of giving back with my microscope. My microscope helps me give back.
My final comment before Rich picks up to talk about how our listeners can help you and contribute to the cause is, we interviewed Dr. Leo Shea III, probably the leading tick-borne illness neuropsychiatrist out of New York City. And he believes that all mental health issues are connected to some physiological condition, right? And he has worked with countless doctors and patients and been able to do a lot of work in connecting Lyme in the brain to psychological conditions. So there is a lot of other work being done there that we have another podcast episode on too that people can listen to if they're listening to this and want to learn more about Lyme and psychological conditions. And from there, I think Rich wants to jump in and talk about quickly how people can help you, Dr. McDonald, in your efforts.
So, Dr. McDonald, you spent your entire career on the outer edge of the Lyme establishment, and we have on many occasions argued, not only on your podcast, but when talking about you, that you were always ahead of the curve, but it was hard to stay ahead of the curve because it's difficult to get money to support the research you're doing when you're so far ahead of the curve. So you were right 30 years ago, and we're sure you're right now, but the traditional research sources of resources are certainly more interested in funding mainstream research rather than, I'll call it, McDonald research. So talk to us about how we can help as a community, and how we can further the advancements that you're making before the mainstream community is prepared.
Well, I was committed to spend about $50,000 of my own money out of inheritance. I spent money to buy equipment and get set up, and I'm glad I did that. And I got additional help from a fundraiser, which is a GoFundMe, around $20,000 for getting supplies, and that was way back 7-8 years ago. What I'm trying to ask for people who are interested in supporting is to get money just for the cost of publishing the papers, because I have all these papers ready to go. And in order to get success in publishing, you go through the peer review, they say, yes, it's ready to publish. And now, in order for us to publish it, we have to get $4,000 for each paper that you publish. And so I don't have the extra $4,000 times 6 or 7, so I'm looking for help from the community to see if they might want to send $25 or $50 or something to the GoFundMe, just for the publication costs, to pay for the publishing. So I can get some of these published, because if it's not published, it's not credible. And to me, it's credible without being published, but for most of the people in the world, they have to see it published before it's credible. And then once it's published and credible, then it helps patients out there who need that evidence to get the medical care that they're seeking and are being denied because it's not published. So that's where I am, and I have a GoFundMe site, and I would let people know about it if they're interested in sending a small contribution, and then over scale, maybe I can get some money to publish these articles.
Dr. McDonald, we're going to make sure that we put this information of the GoFundMe page into our show notes. We're going to make sure that we do promote this as well on our various social media, so we can get more attention to this, because it is really important that these really important topics be made part of the public record. And we did talk last time about the importance of the research data being available, not only for the purposes of moving forward with the research, but giving clinicians the protection they need when they're doing the work they're doing. Because if we don't have published research to support what clinicians are doing, it becomes pretty easy for medical boards to attack the license of doctors who are deemed to be acting outside of the acceptable medical practices. So one of the best ways of, I think, protecting the clinicians who are willing to do the work we need them to do for us is by making sure that these papers get published. So there are very important reasons, but we're going to talk about it from a selfish standpoint. We in the community can't complain about doctors who are trying to help us being put in a box and not being able to help us if we're not willing to spend some of our resources to help change the way that these papers are published and the information is made available, so it gives cover to the clinicians who are willing to be more aggressive. So we can't thank you enough for not just spending time with us, Dr. McDonald, but for all the great work you're doing for the community. You're an absolute blessing to us and to our entire community, and we love you, and we thank you for everything you're doing.
Thank you very much. That's so kind. Thank you. I'm happy to help.
Thank you for listening to our special Tick Bootcamp interview with our guest, Dr. Alan McDonald. To our listeners, we have a call to action. First, if you'd like to learn more about Dr. Alan McDonald, please visit our profile on the Tick Bootcamp website. Second, if you enjoyed this episode of the Tick Bootcamp podcast, please share it with your friends by using the social media buttons you see at the bottom of our post. Third, we here at Tick Bootcamp have created a Tick Bite Blueprint that has been inspired by the information that has been shared with us by past podcast guests. We urge you to visit our website at tickbootcamp.com to view our blueprint. Please note, if there are any helpful hints or tricks or changes you'd like to offer to us for the blueprint, we'd be happy to get your input. Fourth, don't forget to subscribe to this podcast on Apple Podcasts, Google Podcasts, or Spotify to get your automatic episode updates of our Tick Boot Camp podcast. And finally, we thank you, our community, for your comments on our past podcast episodes. Please take a minute to leave us an honest review on Apple Podcasts, on Instagram, or on our website. We make it a point to read every single one of the reviews we get. Thank you, as always, for listening.